Proteins, the building blocks of life, molecules inside each and every one of us. But what if I told you that these molecules could be the key to treating deadly viral diseases? The ever-developing nature of modern medicine fascinates me, leading me to explore protein engineering. Hello, my name is Aditi Srinivasan, and today I would like to talk to you about my work in protein engineering. It focuses on the substitution, insertion, or deletion of nucleotides in the encoding gene in order to produce a modified protein that is more suitable for a particular application. For SARS-CoV-2, diagnosis is often too late. However, directed evolution can be changing that in the near future. It is a lengthy process that involves small modifications to a genomic sequence being tested for favorable qualities. This is done by injecting each modified sequence into yeast or bacteria. Using Hyde-Thoropa assays, which are a form of evaluation, scientists are then able to understand which sequences provide the most favorable results, whether that be growth, competition ability, or adhesion to certain substances. After hundreds of careful trials, an optimal modification is selected to pursue the next phases of testing, and the cycle is then repeated. This method has promising applications in treating SARS-CoV-2, the novel virus behind coronavirus 2019. Many patients with this infection currently do not produce antibody responses until after the time in which treatment, therapeutics, and transmission are no longer viable. However, using directed evolution trials, engineers can modify different components of the antibody surface to block viral receptors without the use of antibiotic treatment, specifically the S1 subunit of the spike protein, which is the protrusion off of the COVID molecule. It identifies receptors on the surface of its target cells and attaches using the S1 receptor binding domain. This causes the spike protein to change shape, and thus the receptors on the host cell to change as well, allowing viral entry. These receptors are known as human ACE2. A cure with anti-HACE2 antibodies disrupts the interaction between the virus and the receptor. In other words, if antibodies that dislike human ACE2 can interrupt the virus from attaching to the receptor, it can prevent infection altogether. I created this model to provide a 3D representation of the various conformations of the monoclonal antibody I just described to you. Monoclonal antibodies are promising treatment to prevent viruses from infecting human cells. Using what we learned from this coronavirus-focused research, we can adapt and understand how to use protein engineering techniques for other viral diseases as well. This research has the ability to change lives. Modern medicine has never seemed more fascinating or necessary to me because it has the ability to change our understanding of disease treatment as a whole. Thank you so much for your time. Here are my works cited.